Hi, my name is Raha and I'll be presenting about Practical 1, which is the basic concepts of statistics and variation. The objectives of this practical are to calculate and describe the following descriptive statistic, which is mean, variance, and standard deviation, to understand the concept of correlation, and to determine the relationship between the two variables. The materials that we use in the practical are leaves, ruler, and weighing balance. For methods, the length and width of 10 leaves were measured and recorded. The mean, variance, and standard deviation were calculated and tabulated. The first step were repeated for another 2 sets of different leaves. Lastly, a graph of the leaf length versus leaf width were plotted using Excel. The relationship between length and width of leaves were discussed. This is the results for leaf length, width, and weight for all 3 sets of leaves with their calculated mean, variance, and standard deviation. This is the graph plotted using the data of leaf length and width. Based on the graph, leaf length and width of set 3 and 1 are linear. This indicates that the leaves are grown in similar sizes. While set 2 data is a bit distributed, this might be because of the location where the leaves of the tree were picked. Set 2 leaves data are relatively on the larger side and were picked on the denser forest, while set 1 and 3 were picked at the outer area of the forest. The leaves of set 2 are larger in order to receive more sunlight due to the competition and survival to adapt to the environment. Other than that, the position of the leaves also contributed to the leaf sizes.
Next, I will answer the post-lab questions. For question 1, set 1 and 3 have quite similar values of mean, indicating that, on average, the length of leaves for both sets is relatively close. Meanwhile, set 2 have significantly higher value of mean that indicates the length of leaves in set 2 is longer than set 1 and 3. As for the mean width of the leaves, the value also shows the difference in width between set 1 and 3 that have quite similar values of mean, but as for set 2, it has a higher value which is 9.34. For questions 2, the meaning of variance is a measure of how spread out a distribution is in a data set. Next, according to the computation, even though the mean value for set 2 is 21, the high variance value of 33.06 indicates that set 2 has a wide range of leaf length. Some of the leaf in this set might be significantly longer or shorter than usual. In contrast to set 2, which has variances of 5.2632 and 2.201, sets 1 and 3 both have lower variance values, indicating less variability in leaves length around their respective means. In addition, when the variance value of set 1 and 3 are compared, set 3 exhibits even less variability than set 1, indicating that set 3 leaves length are more consistent and nearer the mean value than set 1. After that, the meaning of standard deviation is the square root of the variance and is a measure of variation of all values from the mean. And for the last questions, it is necessary that distinct set of measurement for leaf width and length are required in order to fully capture the dimensional properties of leaves, comprehend biological variation, make scientific comparison easier, and enable precise quantitative analysis across a range of research domains. In conclusion, application of statistics helps a lot in understanding mainly about how nature works. For example, nowadays, researchers can even count the precise distribution or population of certain species that aid in conservation efforts, since many species of flora and fauna are threatened to extinction due to human activities. Other than that, with the help of technology, application of statistical measurement, especially in biodiversity field, has been easier since many software can do the calculation automatically and result can be obtained quickly.
I'm Nur Adila Shwani binti Muhammad Zamri. We continue for the lab practical 2. Practical 2 is about factors that affect the distribution and abundance of organisms. Population are collections of individuals within a species, whereas biotic factors are the organisms that exist within an environment. Various species interact to form communities. Inventorying species and comparing their size is necessary to studying community interaction. The purpose of this practical is to categorize plant species and monitor abiotic variables. The objective of this lab practical is the quadrat concept aims to enhance observation power, record plant distribution differences, and infer factors affecting organism abundance and distribution from natural observation. Next, for the method, firstly, one 5 by 5 meter quadrat was created for each type of virus. Numerous quadrat samples were taken at various random sites throughout the environment. Then, every tree stand within the quadrat was examined and noted. Lastly, data were used to estimate the population size and population density within the entire habitat. For the material, you must have raffia string, measurement tape, stick, scissor, and notebook. For the result in quadrat 1, the total number of individuals we found is 71 and the highest species is Micropteris linearis. Meanwhile, for the quadrat 2, Clademia hirta is the highest species we found. For the result in quadrat 3, the total number of individuals we found is 41 and the highest species is Cryptomium fortunate. Meanwhile, for the in quadrat 4 and 5, Clademia hirta is the highest species we found. For the discussion, a term of quadrat is a rectangular sampling area are used to analyze species distribution in specific ecosystem. They are typically 5 square meter in size, providing quantitative data on ecological parameters like species richness and population density. Quadrat technique requires standardized size and random or systematic placement. Standardized quadrat is to ensure sample uniformity and precise data comparison across study sites. Meanwhile, random or systematic placement ensure representative sampling, preventing bias and enhancing data accuracy. There are some pros and cons of using larger quadrat. Larger quadrat provide more coverage, more organism sampling per quadrat, and more representative ecosystem sample due to their larger area and distant environmental conditions. However, larger quadrat also can be complex to handle heterogeneity due to their wider range of environmental conditions and habitat types and require more time and effort for surveying. Next, for smaller quadrat, provide precise population density estimate for animals with high densities and are ideal for studying creatures in intricate ecosystems with dense foliage or complex structures. However, smaller quadrats may overestimate sedentary organisms due to sample bias and environmental changes, while larger ones are more sensitive to location and character. The ecologist is grappling with sampling method issues such as bias, sample size, and ethical consideration. Bias sampling can lead to inaccurate population estimate, while sample size must balance feasibility and representative. Ethical standards are crucial for fragile environment. As for the last discussion, microhabitat competition, each effect, species interaction, and geographical heterogeneity all affect the species composition of plants in quadrats. The distribution of species is further influenced by differences in the topography, soil type, moisture content, availability of light, herbivory, and predation. So, in conclusion, quadrats play a critical role in ecology by providing sampling sites, enhancing the ability to observe, documenting plant distribution, and utilizing observational data to infer ecological factors affecting organism abundance.
In chapter 9, I will present about competition. Competition is two species that uses the same limited resources. Competition has three modes: interspecific, which a competition between individuals of two species. It reduces fitness of both. Second, intraspecific, a competition with members of own species. And lastly, interference, a direct aggressive interaction between individuals. One of the examples we can see here is the interspecific competition between lion and cheetah. Interspecific competition may occur when individuals of two separate species share limiting resources in the same area. Since both species feed on similar prey, they are negatively impacted by the presence of the other because they will have less food. However, they still persist together. Despite the prediction that under competition one will displace the other, in fact, lions sometimes steal prey items killed by cheetahs. Potential competitors can also kill each other.
My name is Caroline Cherida, and today I will discuss Chapter 10 about predation. Predation happens when one organism kills and eats another organism. This will contribute energy to prolong their life and reassure the reproduction of the predator to the disadvantage of the prey. Okay, the examples of predation is Dionea muscipula, or known as Venus flytrap. Venus flytrap is a perennial carnivorous plant and native to subtropical wetlands, primarily in North and South Carolina. It also has unusual habit of catching and digesting insects and other small animals. So in the video, we will see how it traps the insects. Each lobe of the trap has 3 to 4 trichomes, which act as sensory hairs. When an insect contacts with these hairs, it will trigger the trap to close. If the prey continue to struggle, it further stimulates the hairs, causing the lobes to seal more tightly until the insect dies.